Hi, everybody. Thanks for coming. I'm Ryan Scanlon. I'm the head of the viral gene therapy business for Lonza, where we offer contract process development and GMP manufacturing of viral vectors and virally modified cell therapy, including stem cells. I wanted to thank ARM and the conference organizers and all of our panelists for joining us today. Obviously, the title of the conference is Stem Cells, but today we are talking specifically about gene therapies and viral vector gene therapies, and I'm sure everybody may be aware that stem cells or certain types of stem cells are modified using viral vectors, so that's the connection there, and obviously a lot of ARM members are gene therapy developers. So it's really exciting to have this time here at the conference today. I'm just going to frame the discussion with a few opening comments about the topic here. I should say the topic here is on innovation of process development and GMP manufacturing for gene therapies. And then I'm going to let the panelists introduce themselves. We'll jump into some questions, and we'll keep it pretty informal. If you have any questions, feel free to raise your hand and wait for one of the microphone runners. You'll be able to ask a question. We're not going to wait for questions till the end. You definitely feel free to ask some questions. We'll keep it sort of casual in that sense, and really hope to get a nice discussion going here. I think it's a very exciting time, obviously, for the gene therapy world. We're on the verge, hopefully, of seeing several products come to market over the next, let's say, 5 years or so. And I think it's a very appropriate time to stop and think about where is process technology for manufacturing viruses. I think it's starting to be a little bit cliche, but I think it is relevant to think about the comparison on how the mammalian cell culture industry developed through the late 80s to now, and many of you are well aware that the industry started in adherent roller bottles into the late 80s, into the 90s, and then when monoclonal antibodies came on the scene, they obviously had and have great blockbuster potential, and those products drove massive volume requirements, and it was that need for volume that I think really stimulated the innovation in process technology. Companies really started to focus on, okay, how do we get our processes more productive? How do we get yields up? How do we get our processes more robust and scalable? And so then through the late 90s to now, we have a very robust industrial infrastructure for manufacturing mammalian cell culture products, specifically monoclonal antibodies. So then if you think about where are we now with viral vector manufacturing and gene therapy? We're a few years removed from the Glybera approval in Europe. Obviously, there's all the fantastic clinical data on CAR-T, the great data from Bluebird, on the verge of seeing Amgen's product make it to market. We're just days removed from what I guess many consider landmark phase 3 data for Spark's AAV product. And I think it's clear, based on those products, we're definitely well into the commercialization phase for gene therapy. And if you look at the pipeline, it's not just small products, small volume products like Glybera, but there's definitely very ambitious products coming through the pipeline into development, and are going to require massive volumes, or at least I should say in the context of what is massive for viral vectors. So I think we should stop and consider where we are on the continuum. We have a lot of experience here on stage, and I think most people here would agree we're well into the development of process technology here. We're not at the beginning, and many of the folks here have spent their life's work working on developing innovative process technology, but we have a long ways to go, and we should stop and think about where are we. With that said, I'd like to allow all of our panelists here to introduce themselves, and then we'll get into the questions. And again, if you have any questions, please do feel free to raise your hand during the session.
Thank you, Ryan. I'm Richard Snyder. I'm the Director of Biotherapeutic Programs and the Director of Florida Biologics at the University of Florida. Florida Biologics is a CDMO specializing in advanced biotherapeutics, including cell and gene therapies. Our client base is comprised exclusively of private, mid, and large publicly traded companies seeking our services, and we're staffed with 117 seasoned professionals dealing with these complex biologics. Thank you.
So, I'm Robert Kotin. I'm a vice president at Voyager Therapeutics, which is in Cambridge, Massachusetts, and we're developing AAV gene therapy products for neurodegenerative disorders. Before joining Voyager, I was at the National Institutes of Health Intramural Research Program for 20 years, and like Richard, we've been both working on AAV as viruses and gene therapy vectors for 30 years.
Great. Hi, I'm Alan Moore. I'm Vice President at WuXi AppTec, responsible for cell and gene therapy manufacturing. Started out at a company called Microbiological Associates way back when, moved to Genine, and then most recently, before WuXi, was at Althea Technologies here in San Diego, so good to be back. WuXi is somewhat of a late entrant in the gene therapy manufacturing space. We've been producing clinical grade cell therapies since 2007. We're in the midst of expanding our facilities to accommodate both CAR T manufacturing as well as viral vector manufacturing. And we're trying to decide how best to configure a new 150,000 square foot facility designed to do those tasks.
I'm Anthony Davies. I have worked in the field of cell and gene therapy for the last approximately 20 years. I spent some time in the first wave of gene therapy working at Audentes Pharmaceuticals on an oncolytic adenovirus, which even back in those days we took to multi-thousand liter pre-commercial scale. Then spent some time focused on embryonic stem cell therapies at Geron and adult stem cell therapies at Capricor. A few years ago I founded a consulting practice focused on CMC for cell and gene therapy. And I'm currently acting Chief Technology Officer at 4D Molecular Therapeutics. 4D is a Bay Area company, currently a preclinical stage, privately held company. We hope to enter the clinic as soon as we can with a very ingenious and novel platform of next generation AAV-based therapeutics, which are evolved for specific clinical functions.
My name is Janine Winslow. I'm the Director of Research Analytics at Audentes Therapeutics. I'm a relative newcomer to AAV. I worked first at BioMarin on their AAV project, and now at Audentes Therapeutics. Prior to that, I was involved in a lenti gene-based gene therapy product with Bluebird Bio. Audentes Therapeutics is an early stage gene therapy company, and we are focused on severe orphan diseases. We're preclinical.
Great, thank you very much. I thought we would open it up with a fairly general question about the state of where we are in process technology. So I was wondering if the panel could address what you think are the largest challenges the industry faces right now with respect to process technology. And anybody want to kick it off?
I think the biggest problem that I've seen over recent years with these very novel technologies is the field's reluctance to start at the end. I think there's been too much willingness to rush into manufacturing with unit operations, which at some point will have to change. I was asked at another recent panel, what are the cost drivers here? What's the most expensive unit operation in this field that you would most like to not have to deal with? The most expensive unit op in the field is the one you have to change before you launch your product, or even worse, to change after you launch your product. I think we'll probably get into some specifics for viral vector manufacturing later, so I won't do so now, but there are some real legacy unit operations in this field which are getting baked into clinical grade processes and should not. And that's one of my primary concerns.
Any specific examples you think that are particularly, let's say, unfortunate to get into commercial?
So in the 90s, we were manufacturing recombinant adenoviruses in cell trays, which are sort of about the size of the folder you got when you checked in today, and we were purifying by centrifugation, by either isopycnic or density-based centrifugation. These are two technologies which have no place in a GMP facility making a commercial product. Today, groups are manufacturing AAV, adenovirus, similar viruses, not quite using cell trays, but using planar, labor-intensive, open cell culture systems, and purifying using centrifugation-based methods. I think it's a bit depressing, frankly, that 15 years later those same mistakes are being teed up.
Well, that's not universally true. There are suspension systems for making AAV that are robust, and even a little lab at the NIH was able to do 500-liter processes. Now at Voyager, that can be put into a GMP-compliant process that yields reliable, high-quality vector that makes it through a validated or qualified analytics for clinical development. So sometimes we've learned from this.
I agree. I mean, I think it's great that there are some suspension systems being developed. I just think there's enough not being developed that should give the industry pause, and the industry should look to examples such as you're raising.
And it's also probably worth just considering for everybody that the vector, the state of the vector, the diversity of the products out there is substantial, right? You've got AAV, you've got lenti, you've got all the oncolytic viruses and adeno. So, in the case of AAV with backload, I think there's a clear sort of pioneering example of maybe a first generation industrial sort of framework there, right?
I think one thing, if you go back to the analogy to monoclonal antibodies, the industry really encompasses both the manufacturers and vendors of these chromatography systems, et cetera, and bioreactor systems. And there hasn't been an incentive for those folks to invest heavily in the gene therapy space until recently. And I think you're seeing that there are some chromatography options that are available now or are going to be available very, very soon that may serve as a basis of convergence, particularly for things like AAV, that may be a more scalable option. And I think clearly if we look back at monoclonal antibodies in the early days, everyone thought that they had a unique purification process. And at the end of the day, today, many of those processes are identical. And that's driven by a number of factors: availability of good performing materials, and also the ability or the convergence which occurs when the regulatory agencies approve products and say, yes, this system is going to allow us to license a product. And so we have to keep in mind that there is a broad contribution to the field that is now being encouraged that hasn't been there in the past to some extent.
I think the scale-up issues, at least using the Bacula system that Rob invented, are being addressed. I mean, people are pushing up into single-digit 1,000-liter reactors, 1,000 and 2,000-liter reactors. We're consistently knocking out batches at 400 liters, and the process is reproducible and definitely scalable. For the downstream, I think, as Alan mentioned, I think there's going to be a convergence of technologies that can handle the throughput of these upstream yields that will allow separation of full and empty particles that Anthony talked about earlier is being addressed right now through centrifugal processes, but chromatographic scale-up and improvement in customized moieties that are on these chromatographic resins. I think all of those things, again, as Alan said, working with the manufacturers to develop products and ancillary products that support more robust and high throughput product manufacturing.
So it just so happens that for the drug developers on stage here are all ones that develop AAV, and I wanted to understand, is there a, we know about the therapeutic benefits of AAV. I think those are pretty well, kind of talked about. But are there any manufacturing benefits to focusing on AAV as a product type? I guess maybe Anthony from.
I think one of the basic attributes of AAV is these are depender viruses. These are viruses which are not viruses as we understand them in the conventional sense, and that can be exploited by a multitude of methods actually to put the virus together. You can put it together using plasmids, you can put it together using baculoviruses, you can put it together using simplex. There's a number of ways of solving this problem, and although most of us on the stage here have our favorite method of doing this, I think all of us should admit that the field is strengthened when there are multiple options and there's some sort of competition between these methods. AAV, it's relatively speaking, it's small and it's simple. It has. I'm a strong believer that everything you want to purify in biologics has some biochemical or biophysical attribute that you can exploit to facilitate its purification. Ligand-based purification is available. An interesting analogy with the antibody field is, why don't you use an antibody to purify an antibody? It's the perfect specific reagent. Nobody really does that. And the reason is you end up, what that's called is developing two drugs. Okay, one drug to purify your drug, and then it's a sort of ad infinitum problem that you make yourself. Personally, I'm a big fan of simple processes which exploit the basic biophysics or the biochemistry of what your product is, and I think AAV is a really interesting particle in that regard. It has some interesting properties which will lead to some, in addition to the ligand-based methods, will open the door to some simpler, cheaper.
Sorry, just a quick follow-up then from 4D's point of view. I'm not sure if you can share, but is there any thought? Does any thought go into manufacturability in discovery, or is the focus really on product attributes?
We do think about that a lot. We have a fairly evolved view of what constitutes manufacturable. Obviously, I'm not going to disclose technical details of it, but just by reference, companies like Genentech, throughout the development of a product, if the cost basis of the product gets seriously above 10%, that can be a developmental deal killer for that product, whatever it's doing in preclinical work or clinical work. And I think that maybe in the genesis of gene cell therapy, that might be too stringent a criterion. I think it's something, the cost basis, the rules of thumb up there in South San Francisco are, don't let your cost basis rise above 10%, and don't let any individual contributor to that cost basis, any single raw material or consumable, rise above 10%. There's no point developing a drug that you can't afford to manufacture.
Sure. So there are a couple of points. One is talking to CEO of Genentech, Frederick Riva, who recently we developed an immunoligand for AAV9, single-step column purification. LifeTech is now commercializing it, licensed it from the NIH. And what Frederick said, what's the good of having the best vector in the world if you can't produce it? And that goes for any of these new capsids or everything else. And Anthony's absolutely right. So you'd have to have a simple, cost-effective, economical process for doing it. But slightly different than Anthony's take on things. what I try to inform people who've been in the small molecule world is that AAV is very complex. There are a lot of moving parts. Manufacturing the virus requires temporal expression of cytotoxic proteins and replication of a single-stranded DNA genome, which doesn't occur anywhere else in nature but in the Parvoviridae family. So there are a lot of components that have to come together in these producer cells, and it's not just secreting a recombinant antibody, which is very easy to do. So the assembly of the stoichiometric structural particles, packaging this genome, and everything working on time in a large volume is very difficult to have organized for the virus evolutionarily or for us synthetically.
I couldn't agree with you more. I mean, there's the temporal order of expression of various viral and cellular components that have to come together. But to answer your original question, which is what distinguishes AAV from other classes of vector in terms of production, I think you turn to the physiochemical properties of the virus. The virus, since the 60s, has been characterized as one that can withstand heat. Momentary heat up to 60 degrees Celsius, sustained heat at 40 and 50 degrees Celsius. It's resistant to organic solvents. It's a very hardy entity and can be purified and handled very much like other traditional biologics. That's different than adenovirus, which is actually quite a bit more fragile. The fiber protein on the outside of the vector can easily be knocked off. You have to get into special formulation and handling of those types of vectors. Lenti and retros that have envelopes are also very fragile. Present their own issues in handling and purification. So I think as a vector system and its manufacturability of AAVs, I think it stands head and shoulders above some of these other classes of vector.
And compared to Lenti, would you say it's maybe a little bit more accessible or straightforward to manufacture? Not to say it's straightforward, but...
I don't know. I mean, there's nuances to manufacturing all of these different vector systems, and to match the manufacturing with robust analytics is equally as important. These are very complex drugs that we're manufacturing, as Rob said, lots of moving parts, and to be able to characterize and then better characterize. It's an evolution, I think, that the field is going to have to undergo as manufacturing gets more robust. The impurity profiles and the other things that present themselves are going to evolve.
I think to that point, when you compare, AAVs are more simple. It's probably the simplest of those mentioned so far. A simpler virus. Also, in terms of, so how do you better produce it? And the analytics are very important from this perspective when you compare Lenti to AAV, for example. A typical, we call it infectivity ratio. So that's for every particle, how many infective particles are there? With a Lenti vector, you're in the realm of hundreds. With an AAV vector, you're in the realm of thousands. So you need many, many more particles with AAV to get one productive infection. So you have to consider sort of how well the virus works from a biological perspective. And then in terms of producing it and to the point of a simple production system, a simple purification system, the strategy that we like to think about is how do we make more potent virus to begin with? Can we use our upstream process to make more full, for example, virus? And then to that end, do we need to invest in understanding the producer cells and what's going on in terms of the AAV biology so that we can make more infectious particles, so that we can improve those ratios? all those will contribute, I think, to moving scalability and therefore flexibility in terms of diseases that can be used, that AAV could be used for.
Yeah, and I would echo, the analytics is something that is very important, because in an environment where you have regulatory incentives to move products very quickly to late stage and commercial applications, which exists now for a number of these products, you have to be able to advance the development of that optimized second generation process, and you can't really do that without good analytics. And I think one of the points that Jim Wilson made at Penn was that there really needs to be an emphasis on the analytics for AAV, and you talked about the disparity between, you just titering product at 4 different locations gives you 4 different answers, and demonstrably separate answers. I think the other contribution of the good analytics is that it allows you to install them early in the development process, so you can actually identify which vector is best suited. One of the questions which I anticipate might arise as well is, do we need to focus on the vector donor interaction in a particular indication, or for instance, if there's a targeted cancer and we see variation of transduction efficiency from patient to patient, do we need to take that into consideration early on? And that asks a question then of, do we employ big data and genomics in that analysis as well? And all of those contributions of good analytics speed the 2-stage development cycle.
Sure. No, and I think speaking from the CMO point of view, we're always encouraging the importance of to our customers of developing analytical methods and not waiting till it's too late to qualify and validate your methods. And so I just wanted to shift and talk a little bit about the role of outsourcing in the viral vector manufacturing business. I guess first I'd like to address the fellow members of CMOs here, Richard and Alan. What are some of the reasons that you feel that outsourcing development and production of gene therapies is attractive in this space? I guess, Richard.
I think it's expertise. Again, having an organization that can function on all levels in terms of whether it's upstream and downstream purification, whether it's the formulation, final fill, the analytics, certainly quality and regulatory aspects, and even have frontline technicians who know how to handle these very complex biologic entities, handle them without killing them, delivering a potent product at the end, I think is one thing that at least our clients turn to us for.
I used to work for a fellow by the name of Jim Fenno, who was an ex-Amgen regulatory affairs guy, and he likened a contract manufacturer to a big city hospital emergency room. You get to see a lot of things. Not all of them are pretty. You learn from those programs, and you collect the things that work. And I think you guys will agree that you often have a standard which is established by the folks that come in and audit you and what their expectations are. And that helps the next program to go through the door. In terms of the value of analytics, one of the things we're emphasizing is the ability to build those analytics, those cell-based assays, those potency assays alongside the development and the manufacturing activities, and that has the value of speeding the entire process and allowing you to kind of forward integrate when you're under that regulatory incentive to move quickly. I think realistically, many of the indications for these products aren't going to require huge manufacturing plants, and the shared overhead of a contract manufacturer can be quite valuable to those types of products, and in some cases it may be the only economical solution.
Sure. I just wanted to pause for a second and ask if anybody in the audience had a question. Oh, there's a hand over here. Oh, great. Thanks, Sarah.
Thanks for an interesting discussion. So the question I had for you all was around something that it's a bit of an elephant in the room, but I'll put it out there to be provocative, which is while there's been tremendous progress in scalability and transduction and efficiencies in moving what was seen as a very academic and cottage industry to something that's scalable on viral vectors, I personally don't feel as enough scientific effort has been made on the risk side around genotoxicity and insertional mutagenesis and all the other things that are now going to become a reality to deal with if we believe on the promise of products that are coming through. So I'm just really curious in the panel, is that just my own bias and perception that enough progress hasn't been made scientifically there? Just as experts in the field, I'd like to get your flavor on that.
Alan, or?
Is that us? Yeah, I think that may still reside as a challenge that the industry needs to address. I'm not sure that there has been a concerted effort to address that across the industry, and that may be something that a consortium would benefit. It also, I think, points to pulling in additional expertise. We've had a very preliminary discussion with some colleagues at Nextcode Genetics and ask the question, are there tools which we can employ to address insertional mutagenesis? If we've identified, or we can point to 600 tumor suppressor genes, can we put together an analytical package that would help address that? I think very much as the industry did in the early days with recombinant proteins and monoclonal antibodies, I think Merck went out and kind of addressed much of the concerns about the potential for risk there by gene expression, and cloned and sequenced an excessive number of products and kind of threw all that data at FDA, and FDA said, okay, we agree, that seems like a low order event. That may be something that a consortia could do, and certainly the cost for many of those tools has been reduced dramatically.
Yeah, and I don't consider myself an expert on the exact history of the mammalian market, but I know that the first monoclonal antibody was launched in the 80s, and you had Epogen in the late 90s or late 80s in roller bottle, and then it really wasn't until you had a Rituxan where there was a clear blockbuster and a clear volume driver, and I think the early process innovation came from the likes of Roche, and they had their own cell lines and their own process technology, and so did Amgen and Biogen, and then Lonza really made the effort in the mid 90s after the Celltech acquisition, and we really invested once it was clear there was a market. So I think from the CMO point of view, I think there's going to be a lot of progress that gets made, but we're just at the beginning of that front end of it, and everybody's sort of waiting to see when will Novartis get to market with CART19, when will the others make it to market, when will Bluebird get to market, and what really will be the volume drivers, what really will be the criteria that drives value. Is it productivity, is it robustness, is it more product quality attributes, right? And I think we're all sort of waiting to see the final answer, and I think we know it's sort of all of those things, but it's going to be interesting to see how the market evolves over the next several years. Any other thoughts on that question, or any other questions from the audience? Well, so I asked a question from the CMO point of view. I wanted to turn the table and ask from the drug developer point of view. When does it make sense to invest your own capital and fixed cost in setting up your own in-house capabilities? I know Voyager, you had your announcement with your partners in Massachusetts. Rob, can you comment on sort of why that sort of made sense?
Yes, it made sense because Mass Biologics, which is a division of the University of Massachusetts Medical School, the director Mark Klempner is executive vice chancellor and a professor of medicine, has a long history in the production of biologicals going back to the end of the 19th century. The state of or Commonwealth of Massachusetts decided to invest and expand their investment in vector manufacturing, and because Voyager is in the state, and we also had an interest in developing clinical grade manufacturing, we were able to come to an agreement that gives us access to the facility, a new building in Fall River, Massachusetts, where the MBL, Mass Biologics employees and Voyager employees work together to co-develop the process. As Richard had said, it's experience that's critically important for getting these processes done on time and being assured that the product will be clinically useful when the campaign is finished. So by working together with with Mass Biologics, we took the Voyager staff, which has a long history of AAV manufacturing, to work with MBL team to develop a large-scale GMP process. Whether we continue to continue this relationship or build our own facility, as Alan says, it depends on our needs and our capacities, and other CMOs are learning the process and becoming more proficient at producing vector as well.
Can you share any of the thought process that goes into, will you build and try to commercialize yourself, or would you look towards partners for that?
Well, it's an internal debate we're having. I think it depends on how many programs, how many campaigns per year need to be done, and it's relatively, because manufacturing AAV doesn't require a space as large as this auditorium. Can be done in a very small, clean room with the right class air for the different stages of the upstream and downstream. So we haven't made that decision yet. Laboratory space in Cambridge is getting harder and harder to find, so that's part of enters into the equation.
Anthony or Janine, any thoughts on?
Yeah, I mean, I think to emphasize what's already been said, the reason to use a CMO would be for expertise and ability to execute quickly. So you have to have people that, as has been said, it's a really complex thing. You're asking cells to produce virus when they've got their own mechanisms to kill viruses, right, and get rid of them. So what we're asking these cells to do is really quite tough, and it's a fine line between the cells dying and the cells producing enough virus for you to work with. And so in terms of when do you outsource versus invest in your own facility, a lot of it depends on how much material you need, what is your disease target, and what is your route of delivery. An IV delivery is going to take a lot more material than some of the ocular programs would require. So that all is going to inform which way you go, as well as your pipeline. If you're going to invest in not branch out into other types of viral vectors, stay strictly AAV, then it first makes sense to find people to quickly get some virus made, and that's sort of the common model to get your preclinical studies going with material made from someone else. And then the big question is always then what next? How do you get up to the next scale? And that's the hard part, I think, for a lot of people. You can maybe get into phase 1, what about phase 2 and commercial? And that's where it becomes more challenging, and that's where I think more investment into production technology is going to be needed. So, in terms of weighing what's the best way to go. You have to ask yourself how much material you need, how much investment in production technology do you think is going to be necessary to get this to the scale that you need, and then, you know, can you do that better in-house than outsourced?
Sure. I think it's interesting also to look over to monoclonals, to a much more mature, commoditized industry. Genentech has, well, certainly in combination with Roche, Genentech has more thousands of liters of production capacity probably than any other monoclonals producer in the world. As of last year, they also have over 100 open accounts and contracts with CMOs. Okay? Even though they're bigger than probably all of those CMOs combined, they still regard it as a great risk mitigation strategy. It's also worth looking to the field of cell therapy, and specifically the CAR-T field, which is now really well capitalized to the tunes of, you know, multi-billion-dollar market caps. I'm not singling these two out particularly, but both Kite and Juno are building their own in-house capabilities, and they are using CMOs. And I think the question of either or is probably going to become a lot more blurred as years go on in the cell and gene therapy field. And the reason I think that is because it's already extremely blurred in the field of biologics and small molecules.
Sure. And I guess a lot of it has to do with risk mitigation, and
Yes.
you probably, for the big products like Juno and Kite are producing, they want to have some dual source strategies. I'm sure. I'm guessing. And so I guess I want to ask, how do you think the overall regulatory environment plays into the risk of commercialization, and what thoughts go into commercialization with a CMO versus in-house, given the regulatory environment? Is there a certain fear that you'd rather manage that yourself, or would you rather look to a partner that's done it before in the biologic space?
Yeah, and actually on the regulatory front, I'd like to go back to the analytical questions that were being asked just briefly. First of all, I think it's great that this discussion veered immediately into the analytical world. If there's anywhere we really need innovation in process development and manufacturing of gene therapies, it's in none of the above, it's in analytics. And I think I'd also like to, from a regulatory perspective, draw attention to the ARM initiatives which were discussed yesterday for producing reference standard material, and encourage everybody interested in this field to look at the ARM-mediated interactions between NIST in the United States and the FDA, and there are also similar initiatives emerging in Europe between the regulatory jurisdictions there and NIBSC in the United Kingdom. I think this is in many ways, this is somewhat of a safeguard for the industry from a regulatory perspective, to develop strong characterization methods and strong reference standards, and for the heavy hitters in this field to step up and establish minimum standards. I think will be very protective to the field. I think the regulatory implications of going with a CMO versus in-house. Everybody loves to control, except that it's a lot of work to control. I think we will see a lot more in the next few years as BLAs are filed in this field. I think I'm very interested to hear the CMO perspective on that myself.
Sure, and I think from should state I'm not a regulatory person, but from what my general understanding from Lonza's point of view, there's a big difference between ex vivo autologous cell therapy and making an in vivo viral vector gene therapy. We publicly had our deal with Celadon, and I have to actually just give the quick sort of attribution and kudos to Celadon and all of the effort they made to go after a big indication. Where we had the opportunity to work with them to scale up to 2,000 liter scale, and in that facility, while we didn't get to build it, looked way more like a modern 21st century biologics facility than it did a roller bottle facility. And the approach with them is the regulatory guidance is pretty clear. This is an injectable biologic. there's viral vaccines on the market with guidance. So for an in vivo viral vector gene therapy, we didn't approach it too differently than we would a monoclonal antibody, and obviously given the specific special considerations with it being a virus. But I guess I'm actually curious maybe to hear from you, Alan, from the lenti point of view, where it's used as a raw material for cell therapy, how do you view that regulatory perspective?
Yeah, I think our thoughts are that all of the existing guidance and advice and practical kind of applications that C and CGMP is applicable to these products, whether it's the vector that's going to be used with the patient-specific product, or whether it's a vector for in vivo treatment. I guess the challenge that we see, and as we're structuring facilities and looking at how to best support the broadest spectrum of vector development, is for Chinese hamster ovary based products, much of what has shaped the quality control paradigms and the regulatory approach has been failure. And we haven't experienced those failures at large scale. I think, if you look back and you think about viral contamination in monoclonal or protein therapeutics, all of the large scale developers have had some issue or have taken extensive measures like BioMarin to build in sterilization processes for all of the incoming raw materials. That was an outgrowth of that recognition caused by specific events. We haven't seen that in the gene therapy space. That, I think, is one of the challenges going forward. But as you pointed out, Ryan, all of the rules that govern aseptic processing and the production of these products relates quite well, and the vaccine proximity, the safety and QC paradigms are I think well understood. The challenge and the thing that keeps me up at night is which of the various approaches and platforms are going to win out and be the standard going forward, and can you build enough flexibility into the system to deal with that and your facilities. That's.
I have another perspective on this. Our clients are looking at AAV in particular as a platform for multiple indications using AAV vectors. And I think once the regulatory filings are submitted, after they've entered into their clinical phase and they're pushing towards commercialization, they want to bring in product after product right behind that. And once they've created that framework for themselves, really fill that pipeline with the product. So from that perspective, I think everything the other panel members is saying sets that framework for a really robust pipeline of products that fit on that platform, whether it's a vector platform, a regulatory platform, and analytics for that matter.
Just with the pause again, any questions from the audience? Feel free to raise your hand, realizing maybe you can't see everybody over here. Don't be shy.
I actually have a question, if no one else. Do you have one?
Oh, sorry.
Quick question on, with regard to process development, is it serotype specific?
That's a good question. So the particles do differ by surface charge, depending on the genome density and maybe even stability and other things. And if you're using an immunoligand, that certainly narrows down the cross-reactivity or specificity. So it's often there are some generic processes that could be used, but for large-scale production, pulling vector out of 500 liters or 1,000 liters requires a fairly specific, serotype-specific and robust process, and there are common elements like ion exchange or whatever that could be utilized, but to get the most fold purification and concentration usually requires some scouting of media.
I mean, I think from an analytic point of view, the serotype plays a very important role. Janine earlier was talking about differences in particle to infectivity ratios. And my perspective on that is that those are artificial. If you take the laboratory strain AAV2, which obviously was isolated out of laboratory cell lines, it infects cells in culture extremely efficiently. And you have a very tight particle to infectivity ratio, sometimes as low as the mid-10s, so 20, 50-ish. You take a vector like AAV8, and you sprinkle that same vector on those cells, and you'll see a particle to infectivity ratio of 10,000. However, if you put that same vector, AAV8, side by side into animals, an AAV2 vector and an AAV8 vector measured by particles, so equivalent number of particles, the AAV8 will blow the AAV2 out of the water in terms of its transducibility. So you're getting an artificially high particle to infectivity ratio in these analytics. That's a place scouting cell lines and other enhancements could bring that particle to infectivity ratio down. And really, you can get into some product equivalency testing by testing an AAV8 vector on its paired cell line versus other serotypes.
So is it fair to say there's sort of a common theme that analytics is incredibly important and it should be an area of increased focus? I think
the thing to let's look back at monoclonals, then the concept of a well-characterized biological, which I think it's amazing to remind ourselves that 10 or 12 years ago, people debated whether that was even something worth debating. And now I think we're well into the world of biosimilars. So I think the question that you need to ask with something which is admittedly much more complicated than a biological is, are we going to have a well-characterized gene therapeutic or cell therapeutic for that matter? The question we should be asking ourselves is not a question, is that we need to have an adequately characterized gene therapy or cell therapy. If the particle to PFU ratio or the solid hold, empty to full ratio, whatever it is for your flavor of virus, if that is genuinely a critical quality attribute of your product, well, first of all, you need to determine whether it is, whether they will assume the empties are the impurity. Are they having a material impact on clinical efficacy and the tox profile of your product or not? If they are, you need to get a handle on it. It's probably going to be the most controversial analytic for this field. Infectivity assays have been notoriously hard to pin down in the entire history of virology. Working on recombinant adenovirus in the 90s, we validated an infectivity assay for that virus and spent well over a year crunching away at the error bars on that assay, and we got them to where it was within pretty much plus or minus a log, which is terrible. Took it to the FDA, and the FDA said, wow, we've never seen anything even near as good as that. Why do you do all that work? It would have been, we don't really believe it's a critical quality attribute. So I think that's those sorts of lessons need to be looked back on, and we need to adequately characterize these products.
And to this point, I think a complicating factor with gene therapy, well, with AAV specifically, and others as well, but is the component of the immune response in humans, because it's something that's tough to mimic in animals. You can do it, it's not always predictive. And so, one of the problems with AAV is how big of a role does this immune response play on inactivating your virus, and that would affect your dosing. How do you model that? And to some extent, we're going to rely on clinical trials and what we see based on what's done in humans. There's only so much you can do in animals, and then you really have to go see what happens in humans. Yeah.
Oh, I know there was a question in front.
Sure. This is a question for the drug developers on the panel. And given the current state of the art, how much of an issue are pre-existing antibodies to AAV? Do you think about this in terms of the therapeutic efficacy over time, or patients who may be excluded from therapy? That's kind of an issue that hasn't been touched upon yet in the panel, that is pre-existing antibodies to AAV.
So that's a really important topic, and it's a really complicated one. One of the complicating factors is that the assays to detect pre-existing antibodies are, every lab sort of has their own assay, and so one person's 1 to 10 or 1 to 50 titer is not going to be the same as another person's. And so when you look back in the literature and try and compare pre-existing immunity in any of the trials that have been done, it's hard to correlate the data. So that's one of the one factor that's difficult. Another factor that's difficult is how much is too much. We do know that pre-existing antibodies can affect the efficacy of your dose, but it's a big kind of murky box because how much is too much and how much do you need exactly to we know it can be overcome with a larger dose. We also know larger doses have immunological consequences. So it's a tough question, and part of the reason it's tough is you can't do so much in animal modeling. And so I think a lot of companies are struggling with how many pre-existing antibodies are too many or too much. How do you exclude patients based on a level of pre-existing antibodies?
Yeah, I think that's a very insightful answer. It's complicated. Mm-hmm. Evasion sounds a desirable attribute, but it also depends on the potency of your virus. Mm-hmm. And this also applies to this question could equally well be asked in a cell therapy session. Okay, can your therapeutic, whatever it is, do what it needs to do in the presence or absence of immunosurveillance? Obviously also has profound implications for repeat dosing. But I think what it boils down to is another sort of angle on what I was saying before, is what the field needs to do is really understand the critical quality attributes of its product, and what is your desired or your needed PK and PD for this product. So I think all of which is why I was saying it's a very good question. We don't have an answer, but we do spend a lot of time thinking about it, but it needs to be thought about in a systematic manner.
So we have 2 minutes, maybe time for one last question if anybody in the audience has a question. If not, I do have one last question, maybe just a quick kind of a fun one. Anybody have any bold predictions for the future of manufacturing of viral vectors?
I think the future is bright, and I recently saw a great presentation which showed a picture of an automotive plant 50 years ago and an automotive plant today. 50 years ago, the plant was full of sort of people in white coats, individually wrenching on cars which sat in the middle of the floor. Today, the automotive plant is unrecognizable. There's not a human inside. It's all done by robots. Picture of a pharmaceutical manufacturing plant 50 years ago versus today kind of looks similar. I think there's a general feeling in the pharmaceutical industry growing very rapidly that there's a lot of unmined benefit in that industry, and manufacturing of pharmaceuticals, especially advanced products, really hasn't tapped into this. I'm sort of cup half full about this. I think there's a lot of low-hanging fruit which can be taken. I think the last 50 years haven't done a good job of it, largely due to regulatory overhang. But I think there's a lot of technological development ready to be done in this field.
Specifically in automation?
Not specifically in automation. I think in ingenuity, in characterization, and in meeting the needs of regulators better.
Sure. Any other final thoughts?
I think, prompted by the factory image, I think one of the things that we may see is increasing personalization of therapies, whether that's the vectors themselves or the combination of vectors in the patient's cells as a therapeutic mechanism. And there is a movement to link the individual characterization of the patient and their potential response to therapies back to the manufacturer of that product. And if you think about the challenges there are many, but every opportunity or every time we see compelling results, we see an individual immunological component contributing in some of the breakthrough therapies that we're funding very heavily today. So that may be a migration that occurs down the road.
Sure.
30 seconds. We have 30 seconds.
Well, I think it goes to the, I'm sorry, cost of goods. As Anthony said, I think Rolls Royces and Ferraris are still made by right-coded technicians, whereas we want to make Civics or things like that. So it's broader markets at lower costs that could be affordable.
Great. Well, I think we have to leave it there. Thanks so much for your time, and thank to the panelists for all of your time and a wonderful discussion.
Thank you.
Thank you.